Okay, we've covered some of the anatomy of our uh, nervous system. Let's talk about the communication in the nervous system. Um, when neurons communicate, we can have presynaptic uh, communication. So we have a neuron generating an electrical signal. When we get to the synapse, which is the meeting place between two neurons, they don't actually touch. We have a chemical reaction between them. So the signal comes down, you get to the end of the neuron. At the synapse, it's going to release a chemical, a neurotransmitter, that's going to diffuse very quickly to the next neuron. And then that uh, chemical signal will be converted back to an electrical signal if there's enough of it. So you have postsynaptic. If there's enough stimulation of that nerve, it will fire and start an electrical signal again. If there's not, it will remain quiet. So electrical up to the synapse, we release a neurotransmitter that moves over to the next cell. And then based on all the inputs that, so here's an illustration of that. We have the presynaptic uh, boot up here. So the signal comes down from the top of the screen. As that electrical signal gets there, it's going to take these vesicles, they're going to fuse with the outer membrane and empty. That neurotransmitter, represented here by the triangles, is going to go to the postsynaptic cell and bind with the appropriate receptors. If enough of those receptors get stimulated, we are going to have a postsynaptic cell response or firing. So the signal will continue on. So you've got the signal propagating down. The two don't actually touch. So we go electrical, and then our neurotransmitters are chemical, and then we're stimulated on this side. If it does fire, it's going to be a electrical signal again. So how are signals moved along the axon? So we have from the body, we have the axon that's going to carry a signal. There's 4 different states we need to observe or see as this electrical pulse moves by. So we start at a resting state, then we have a sodium flux in. So sodium is our extracellular cation. It's going to gates are going to be open. It's going to flow down its concentration gradient and move inside the cell. Potassium gates are going to open, and then potassium moves out. That's step 3. And then there's the return to the resting state, where the potassium is moved into the cell, primary intracellular cation, and sodium is moved out again, primary extracellular cation. Here's the handout or page that we're going to look at. So you can follow along with this or just watch what I'm going to do here on the screen. We're going to go through each of these steps one at a time. Those 4 steps are represented here in the 4 pictures. So we have the steps where we go. The resting state, sodium has been pumped out of the cell and potassium is pumped in, and then we have our signal get starting. So we have some sort of signal. So normal pump, we have a little more potassium inside than sodium outside, so there's a difference there. So we have the depolarization. It's a stimulus that's enough to start to generate the signal along the axon. The first step of that axon firing is the opening up of the sodium channels. This allows the ions to flow out. We produce a net negative charge um, on the outside because we've got all those positive charges moving into the cell down the concentration gradients. Open up the cell. Sodium goes down its concentration gradient, takes positive charges. Now we got more positive charges than inside than outside. So you're going to change the polarity of the cell membrane or across the cell membrane. So the stage is called depolarization. So we've stopped there. So beginning of the repolarization. So the gate on the sodium channel closes, the gate on the potassium opens up to allow everything to flow out and balance out. So you're trying to reach equilibrium. The sodium is rushing in, potassium is rushing out. We have your gates. This one starts closing, this one's open. We have a balance or a do-si-do kind of thing of our positive charges. So we've changed the positive charge on the outside versus the inside. State is the repolarization, so we have to put everything back where it was to restart, get it back in the resting state. So all the potassium has got to be sucked into the cell, all the sodium has to be pushed outside the cell. So we get back to that resting state. Once we are back in that state, we can accept another signal down the axon. Until that point, the neuron is called refractive. It can't respond to that. So the end of the nerve impulse, the gates close, the sodium and potassium ATPase pumps put everything back where it's supposed to be. So they're going to pump the potassium inside the cell and get the sodium outside. So that'll be done by the pumps that are running more or less constantly on the cell membrane. So there's that discussion. Again, we have the resting state. Sodium rushes in. It's going to change where the positive charge is. We're going to have more positive on the inside. Potassium runs out. We sort of balance things out in the short, and then we slowly replace one for the other to get back to where we started. So representing this visually, we're going to move down the axon. So first thing you have is sodium coming in, and that's going to flip the charges. So it's called the depolarization. So this is a cartoon cross section of an axon. So sodium comes in, that's going to bring positive inside. The inside is now pulling more positive than sections over here. So the signal starts to move. Once one is excited, it tends to ignite or excite the one next to it. So we start to move over. Behind that sodium influx, we have potassium gates opening up and spewing out in some way, shape, or form. So this is a state right now that we can't take another signal until this resets on the backside. We can't take another signal. This also, the sodium in, potassium out, also sets the direction of motion. We can't move in both directions on this. So with the sodium first followed by the potassium, the signal always has to go from left to right. And we'll continue to do that until everything resets. So you can see that this stimulates the next one over. Potassium is following behind. This sequence here is what prevents things from running backwards and keeps the nerves all one-way traffic, which is the way we want it. So you can measure that change as we move through. So you've got the resting state where we're normally at a negative 70, which is on the inside of the cell. If we have the potassium rushing in, or sodium rushing in, sorry about that, you're going to have the inside of the cell getting positive in some way, shape, or form. So this is our action potential, where a spike as the sodium moves in. Part 3, we need to get everything back sorted to 0. So we've got sending the potassiums out is going to bring that back to where we were. There's usually an overcorrection there until we get back to our resting potential one more time, back to where we started. So sodium going in is going to make the inside more positive. Potassium leaving is going to bring that back to baseline at minus 70. And then we're going to have the potassium ATPase pumps are going to exchange those ions, the positive for a positive, to get back to the resting state. And we're looking at this happens all in, measured in thousandths of a second, milliseconds. So